Hi everyone, so today I'm going to go through a couple of different types of genetic mutations. So these are basically mutations that occur on different types of chromosomes, and there are 7 main mutations that I'm going to go through today. These are deletions, duplications, pericentric inversions, paracentric inversions, balanced translocations, unbalanced translocations, and Robertsonian translocations. So firstly, let's start off with the easiest one, which is a deletion. So before we do, actually, I will go through just the basic structure of what a chromosome is. So, as we all know, a chromosome is usually this X-type shape. And for this particular lesson, I'll say that it has 3 main parts. Firstly, it has a P arm, a Q arm, and a centromere. So, the P arm, what does that mean? P means petit, and therefore in English it's small arm. This is always the shorter arm of the chromosome. Q arm, on the other hand, is the longer arm. And the centromere is the center, or the point at which the 2 sister chromatids cross over one another. And there are many different types of chromosomes. That's why I hesitate sometimes to say that it's the center of the chromosome, because you can have the centromere positioned at different points. So we can have a submetacentric chromosome where the centromere is slightly off from the center, either to the top or the bottom of the chromosome. There can be an acrocentric chromosome where the P arm or Q arm is quite short and the chromosome essentially is quite long on one side, quite short on the other. Or you can have just metacentric chromosomes, in which the centromere is in the direct center of it. But today, I want to first go through deletions, which are genetic mutations that essentially result in the removal of some genetic material. So in this video, I'll be going through 2 main chromosomes, chromosomes 7 and 22, just because their sizes are quite different from one another and they're easier for the future mutations that we'll go through. So we have chromosome 7 here. So say, for example, that in the fetus, some small part of the Q arm of chromosome 7 is deleted. So what does that mean? So say we take this region, for example, and say, for example, this region codes for a specific characteristic. If this region is lost, then that means that the fetus will not have that characteristic, because essentially the instructions aren't there. For example, if you're trying to set up a new wardrobe and you don't have the instructions, you can't set it up because you don't know how to do it. So it's the same idea in the body. So deletion basically just results in the removal of this part, so it no longer exists. Next, we have a duplication, and a duplication is just the opposite of a deletion. Instead of taking away a certain part, you add that exact same part. So once again, taking chromosome 7, for example, if we have this chromosome here, and this particular region, if we were to duplicate it, then that particular Q arm of chromosome 7 will be double the length of that specific region. So what I mean by that is that the chromosome will be this long. Because we've added 2 or another one of these regions to it. Next, a pericentric inversion, is different because it doesn't actually involve any other part of the chromosome in terms of removing it or adding it, but rather flipping it. So, if we were to have a chromosome like this is the centromere, okay? And in a pericentric inversion, one part of the chromosome that includes the centromere is flipped. So, what do I mean by that? So, in this particular chromosome here, say we flip this region. what will happen is that this will go to the bottom and this will go to the top, which will mean that the chromosome will look something like this. All right. So as you can see, the shapes of these two chromosomes is different, but that doesn't make that much of a difference. It's just that because the genetic information has been switched over, it can cause a certain type of mutation in the fetus. So that's what a pericentric inversion is. In terms of a paracentric inversion, it's the same concept, except that the centromere isn't flipped in any manner. It's a certain part other than the centromere that's being flipped. So, for example, if we take this part of the chromosome here that I've circled, if we were to switch that, essentially, it wouldn't make that much of a difference to the actual structure of the chromosome. However, in terms of its genetic coding, because we've changed that, it'll cause some sort of mutation. So, this chromosome here, X-shaped, we take this region, invert it, it'll look the same, except that there will be a change in the trait. Now, moving on to balanced translocations. All of the different mutations that we've gone through so far involve just the one chromosome. So up here we had a deletion, then we had a duplication, then a pericentric inversion, and paracentric inversion. Now the difference here is that in a balanced translocation, there is a swap of an equal amount of genetic material between two different chromosomes. So for example, if we have the 7th chromosome here, so I'll draw them new here, so 7th chromosome, and the 22nd chromosome, we have here. So, say for example, they undergo a balanced translocation. So as I mentioned earlier, there's the Q arm and the P arm. And say that the P arm of the 22nd chromosome swaps with the P arm of the 7th chromosome. Now in this case, because we're swapping both the same arms, even though these two chromosomes may have the 7th chromosome's P arm on the 22nd chromosome and the 22nd chromosome's P arm on the 7th chromosome, there is no change that occurs. Essentially, because we have both genetic material present, even though it's on different chromosomes, there's no ultimate negative outcome because the genetic material itself hasn't been tampered with in any way. So the chromosomes will still retain their shape. However, in an unbalanced translocation, the story is a little bit different. This is because, in this case, a piece of genetic material is swapped between the chromosomes, but it's done in such a manner that it doesn't allow for equal sharing of the information. So in this case, although genetic material may have been swapped between the 7th and 22nd chromosome, it may change the size of the chromosome or the particular genetic material it has in order to have a negative effect eventually in the fetus. So the two mutations that we just covered here are balanced and unbalanced translocations. And there's a final translocation that I'd like to go through. So we've just seen that genetic material has been swapped between two chromosomes. However, there can be different arms, two complementary arms swapped as well. So that's what a Robertsonian translocation is. It's when two Q arms or two P arms of different chromosomes join together. This means that, for example, as I mentioned earlier, an acrocentric chromosome that has its centromere at the end of the chromosome be turned into a metacentric chromosome, in which the centromere is at the center. So say, for example, we have two chromosomes here, as so. So we can clearly see that this chromosome here is an acrocentric chromosome because its centromere is not in the center of the actual chromosome. However, this one is, I would say, a submetacentric chromosome because it's not quite at the end, however, it's not also in the middle. So if these chromosomes were to go through a Robertsonian translocation, what would happen is that, say for example, this Q arm here, I will just redraw that. So this Q arm here swaps with this Q arm here. So what will result is that there'll be a massive chromosome that's quite long, because as you can see, this Q arm is quite long, this Q arm is quite long. If they join together, they will create one massive chromosome that's double their size. So that's what causes a genetic mutation as well. And that can cause other problems as well, because essentially genetic material can be lost, and there can be a scrambling of genetic material because instead of the normal Q arm linking to a P arm, two Q arms are joining together. So those are the 7 main chromosomal mutations that can occur. Deletions, in summary, are just when a certain region of a chromosome is removed. Duplications is when that region is duplicated or doubled. Pericentric inversions are when a part of the chromosome that includes the centromere is swapped or flipped. Paracentric inversions, on the other hand, is when a part of the chromosome is flipped that doesn't include the centromere. And finally, the 3 types of translations include a balanced translocation, which involves the swapping of complementary or genetic material where there's no loss. Unbalanced translocations are when the genetic material is swapped, but is done in a manner that doesn't allow for equal sharing. And finally, Robertsonian translocations are when 2 Q arms or 2 P arms join together, which means that the chromosomes can essentially change their shapes.